So, so the secondary structure generally are predicted by two methods, that is the ab initio based or homology based. So we have seen in other cases also that the mostly now you can see that the tools that are the algorithms that are based are mainly in broadly divided into two categories. That is, either it will be ab initio based. Ab initio based means only the sequence and you will try to predict with the only the given sequence present, or either the homology based. Homology based means the sequences will first try to align the sequences, different sequences, try to find out the evolutionary sequences, commonalities, and then we'll try to predict. So secondary structure prediction is also based on the tools are also based on these two categories. One can be ab initio based or the homology based. So now what are the tools and how the algorithms work? So for ab initio based methods, relative propensity of each amino acid has been calculated, that if the amino acid is present in a particular sequence, it is present in an alpha helix or beta strand or coil form of structure. According to the based on the different experiments, relative amino acid propensities are calculated, and this has been used by in the Chow-Fasman algorithm. You can see that amino acids, different amino acids, either they are what are the propensities. Now based on that, if a sequence is given, so if a sequence is given, so they will see that what is the propensity value of that particular amino acid in which. So after all, the sum or the total value will be calculated based on a particular window size, and if a threshold of 4 or 5 amino acids, particular amino acids is crossed, that it will be remaining in alpha helix, so it will be predicted as that the amino acids are in alpha helix structure. Otherwise it will be classified that it is predicted that it is present in the beta strand structure. So this is how the Chau-Fasman algorithm works. So window size here is set of 6 residues for alpha helices, and there are continuous sequences are needed to decide on it. So here you can see that you can perform this via this web tool. So you can go. So you can see that it has been.
কেমন আছিস বল? কবে আসছিস? কবে আসছিস?
So we will be taking one sequence. We'll try to predict using the Chau-Fasman secondary structure prediction. So the sequences. So one method is the Chau-Fasman, another is using Garnier method. So here we will place the sequence in FASTA format and we'll submit the sequence. So here we can see the residue totals, the turns, helix, where is the turns, what is the prediction, we can find out in the with the single amino acid sequence. We can find out based on the propensity values. It has calculated that what can be the thing. So here we could not see any helix. It is all sheet for this. In that, in this region, it will be it is in turns, and then again in helix, and some part can be in turns. and then helix, and this way it has predicted in this sheet. So there has been no helical structure found here. So we can, by this using this has been done only with a single sequence, and it has not performed any kind of homology modeling. Now another thing is that you can also use Garnier secondary structure prediction, which uses another kind of similar but little different kind of algorithm. So here you can see that Garnier predicts protein structure. It has also predicted some helix structure. So now you can understand that we cannot use any tool blindly. The thing is that these are all first generation tools and algorithms that have been done in 70s or 80s during that time, using the statistical tools when there was not many data was present for the protein structures and their predictions. So now little going advanced, we have now other with the increase in the amount of data, there has been evolved which accuracy gives high, which is accuracy tools accuracy are much higher. So we need to look at this accuracy of each of the tools before concluding that our what will be the structure, final structure of the protein. So now with this, so this has been gone with the job first one. Another is that Diode method. We can use this tool. So here we can see that where we also we need to predict. So this has been done by Garnier oscillation adoption method. This is the Diode method. So the same. So in the same way, we have paste the protein amino acid sequence, and we will click on start prediction. So here we don't need to provide in the master format, I guess. So here you can see that it has predicted that which one will be in alpha helix, which will be in beta strand. You can see in the graphical format it has been predicted. So also the protein name, then the algorithm, and how it has calculated or it has concluded that it will be in the helix position, how the calculation has been formed. So you can see that from the Chow-Fosman algorithm, which did not predicted any helix, we can see that it is predicting helix from the other algorithms. So with all the beta sheet helix and the turns. So in this way we can identify it. So now and moving on, so we, after this is the homology based method. So methods homology based methods are the ones that also takes combines both these methods. It first align from the homologous sequences, and based on that, from the individual sequences it will try to identify the structure, base structure for each individual sequence. Then it will fit the predicted secondary structure onto the alignment, and then it will make. So it goes another round of check. So it goes another round of check, so that so that it goes another round of check where also the multiple sequence alignment is performed, so that so that it also takes in view the evolutionary information on it, and then finally predicts the final prediction of the structure is done. So here, so another is that nowadays neural networks are also being used. so that the accuracy can be improved along with these two broadly based methods. So here we, it helps us, it goes a round of iteration to predict the best final at which the structure will be present. So one of the tool is this Cypred, which uses Cyblast to align and then perform the alignment and then perform the prediction of the structure. So if we go to Cypred, so here we can see that we can also find out membrane helix prediction, also profile based fold recognition. So you will try to find out the predictor secondary structure. So we will use the sequence data. So you know the PDB structure data can also be utilized in identifying the secondary structure, what was that. Also here domain prediction is there, function prediction is there, structured modeling is there. So different kinds of activities can be performed. So we will just paste the protein sequence. Now we will submit it. So now the Cypred is usually uses position specific iteration Cyblast for performing the alignment. So here we can see that different types of strand, extracellular helix, coil is given. So according to that, it is showing that here it is in the helix range, which is in the coiled range. So you can also use in the cartoon format. So which one is this amino acid and which one is in the helix range, which is the coiled range, and the confidence of prediction. So you can see that it has with the like this ones which is having a more deep blue gray color, is that is highly confident of predicting it as in the helix structure, and those who are not so means it's less confident, and the target sequence and everything, and you also can get this PNG download and can look at it. So you can download it also the result. So you can see that how it has progressed, the tools and algorithms, and how it is predicting also with the confidence scores, that you can be of confident that this is how much you are confident regarding it that belongs to this particular thing. So this is how you can go for this Cypred. So another tool is there for this secondary structure prediction, that is the JFred. So this is also a secondary structure prediction server, where you need to paste this, try to make prediction. So see, it will first perform the homology modeling. So while performing the homology modeling, it has identified different structures having the blast T value, like it has mapped with the insulin, then the different other things. So from here it will, so it's similar to those with the known structure. So it is saying that the sequence we have submitted is similar to those. This may provide a, so if our sequence which we provide has been found out in PDB the different similar kind of structures, then it will help us in providing a more accurate secondary structure assignment. So it will not only rely on the prediction algorithm, but it will also take help and the guide from the different these PDB structures. So this will give us more accurate results. So since it can, this is taken, since we have taken from insulin this thing, so it got mapped to it. So you can see the log files also, that what is happening at the current stage. The different finished. So now here you can see the result. So here you can see the result. So for the results, we can see now, we can see that, you can see in HTML version that from where it has been found, identified, and the query and which structure it has taken. And now also the details on the acronyms used. So if you go here, so you can read it whole and you can understand that what does it mean and what the results, how to interpret those results. Lupus 21, Lupus 14, Lupus 28. See, this is wild prediction for the sequence. These are binary predictions for each location, or prediction of solvent accessibility. The consensus prediction, helices are marked as red tubes. JNet confidence estimates, it also gives the confident estimate for the prediction. So high values gives that, it means high confidence prediction. Then alignment based prediction. So one, the different kinds of uh, helices are then HMM profile based prediction. So different kinds of prediction it also performs at a single go at using different kinds of algorithms. And PSSM based prediction. So you can identify all these things. So if you go to the result, you can see that what is then confidence levels. So you can see that it is, some regions are highly confusing even in the range. That it is marked as red tubes are those which are in the helices. So you can see that these regions are present in the helices is showing, and it's in the HMM based profile prediction, that is also quite similar to it, and you can see that this is also belonging to that particular helices only. So in this way, all these things you can identify it, and also you can use in JALView. So if you remember that in JALView we can see the multiple alignment and all during the multiple sequence alignment. So let us, so if we launch it here, so we can see the sequence and all, we can get the same thing in JALView also. So from here also you can find out the different sequence alignment structures, and as well as the different alignment scores and all. So these are the, this was the query, and this is all those sequences where it has got mapped and identified, and from there it has predicted, so you can get the full view like this, which one is helix with all the scores, coils, and all the homology as well as the prediction algorithms. So you can just download it in the format of the GelView, and just you can, if you have installed the GelView application there, you can just click on this and it will automatically get open and you can get a full view of it. So this was another tool using JPred code which helps in the prediction. So now another thing is that these are all for the generally for each protein structure. It is difficult for identifying, but it becomes more difficult in case of the transmembrane proteins, because the transmembrane proteins have a hydrophobic and hydrophilic both these domains, and their presence and their structure. So their structures are generally most like of a beta barrel like structure, because of they form a kind of a having some kind of permeability as well as having both hydrophobic as well as hydrophilic head and all. It becomes much more difficult to identify particular this kind of proteins having a beta barrel like structure as well as a coil like structure. And it is and the algorithms that are generally used for the identification for this alpha helix and beta sheet for a normal protein, it becomes a little bit difficult to identify in case of this transmembrane proteins. So there are different kinds of tools that have been developed to particularly detect this kind of transmembrane proteins. And if you get an unknown sequence of a protein or can identify an unknown sequence of a protein, so if it is present in a, it is a kind of transmembrane protein or not, so you can also predict it by using it. If we go to these tools and try to identify the sequences, and if it is generally it doesn't give this particular beta barrel and coil a certain percentage of it, so it cannot be used as a transmembrane protein. It will definitely be a different kinds of protein. So let us look at these tools one by one. So let us find some transmembrane proteins. Find it. So we let us took this one. So by taking these sequences, so it will need a sequence in the FASTA format. So we want in extensive output format with graphics. So yeah, so you can see that the human number of predicted TMHA expected number of amino acid is this much. First 60 amino acids is not there. So TMHMM posterior probe. So HMM here it is a hidden Markov model to identify. So in the transmembrane, this red is transmembrane, blue will predict about the inside, and violet is that outside of the cell. So here we can see that how it has predicted. So it has predicted to be in outside. That is, so it is predicted it will be staying in outside. So we have to look at it that which is correct or not based on this. So we can try out with another protein. So let us look at it. That's what it predicts. So here you can see that it has predicted in helix, as well as that the number of amino acid number is much more, and so we can see that it has been told that many probability is many for many amino acid sequences to be in the transmembrane and little less to be in the inside and outside. So this is the kind of transmembrane protein we can see from the result. So in this way we can identify the sequences. So using this tool we can identify that if the one protein is a transmembrane protein or not. So another tool is there. So we all can what we can do is that we can check at various tools and then look at the confidence level of those tools, and then only it will be better to conclude, because every tool and algorithm has some of its limitations. So here can give TM filter or the signal prediction. So it helps in predicting also that that it helps in signal or not. Okay, so now so this is constraint consistent topology will predict. So do you all have any questions meanwhile? So we can meanwhile we can look at other two also, but then we can see the result. So here another tool is this. You can see this obvious. So topology and signal peptide predictor, that it is also does the same thing. So we can trace the same sequence here also. We can look at compare the different results. Yes, so it is quite fast. You can see that it is giving that it is a signal receptor. So non-cytoplasmic, cytoplasmic, and its probabilities. So violet is this transmission. So it is the result is quite similar to what we have got in TMF rate. So you can clearly see that here it is a kind of transmembrane protein having some signal peptides available as well. So you can also find out the probability data and download here. So which one is the cytoplasmic, which one is non-cytoplasmic, and all region you can find out. Latest 2046 of results with UNS result. So meanwhile we can look at another using UV plate. So here it helps in this transmembrane beta barrel prediction. So because since you can see that the transmembrane protein generally remains in a beta barrel structure, so it will help us in identify this. So you can get a knowledge about that what are the transmembrane beta barrel proteins. So if you want to know that how the membrane proteins are constituted. So membrane proteins are a very in, interesting thing to for different kinds of because it helps us in understanding different kinds of signal pathways, and it helps in transducing signals from one or to another. So it is of much interest in the research interest. So that's why different tools have been developed to identify if it is helps in signaling or not. So you can get to know about it and also the prediction methods. So if you can see that it uses neural network based approach and some statistical learning and the SVM based approach, and helps in the combined prediction. So it is the neural network as well as some statistical and SVM methods have been combined to identify. So you can identify to achieve the better accuracy. So you can read thoroughly regarding the algorithm as well as which uses using it. So if we submit the sequence, so we can give the protein name. So we can will use the FASTA format. We will paste the sequence here. So now you can see that you have an option that what to use, that only neural network based, one ASVM or combined. So you can also use different kinds of threshold. So we are using the combined. So here you can see that the protein is likely to be a globular protein. So the predicted barrier regions, it is not predicting. So here it is predicting. So now we can also try to find out if we can lower our threshold, but still, so it is. We can say that it can is moreover confidently predicting to be a globular kind of protein. curve is still not given, so we can try with some another protein. These proteins we can find out with insulin that what is it tells. If we run, so it is also using a globular protein. So it is definitely was not to be what we wanted, but still just to see that how much it can predict. So you can just try out different methods to identify using different threshold and different algorithms as well. So if we just change this algorithm as well, so here you can see that it is predicting something. So you can see that if we changing the prediction method, there will be a change, and we are different kinds of things. So based on your, you have to look at the literature also, and also need to identify which tool is giving and which algorithm is giving the better accuracy to conclude for any particular protein. So these are the tools that can help in prediction of the different kinds of the transcription proteins. Now another kind of protein which is of quite particular research interest is the leucine zippers. So these leucine zippers have some specialized wild-like structure. They are belong to this. So for that also we need to have another algorithm and another tool. So this tool helps us understanding that if zippers are there or not. So let us find out some. If we take the sequence of it, so we'll submit. The number of potential leucine zippers, it is saying 0, so it is not rating anything. So coiled coils which does not correspond to a leucine zipper, so it is saying that all the sequences it doesn't correspond to any kind of leucine zipper. So we can try out different proteins as well. So it generally predicts that how many that coil structures are there or not. It is kind of look for those only. Some examples of So here you can see that it is saying the number of potential leucine zippers is 1 and the sequences. So it is a kind of a leucine zipper. So coiled coils we do not is none, but all the so it is a leucine zipper. So we found out that protein only. So you can see that it has correctly predicted it to be a leucine zipper, but other sequences it won't be predicting it as a leucine zipper. So we can try with this protein also. So you can see that here it is saying that it is not a potential leucine zipper. So only those having that particular it can predict. So in this way we have got we can identify different kinds of proteins having different kinds of secondary structures, starting from firstly the helix structure, the beta sheet, the coils, then a particular kind of transmembrane proteins which having a beta barrel-like structure, or the leucine zipper-like domains, or the so we can identify. So also the tools uses two different kinds of algorithms. One is this ab initio based, where the different amino acid propensities has been calculated, statistically calculated based on the different kinds of proteins present and the different kinds of amino acid sequences, and also by the homology based modeling, where it takes combination of both propensity level and also the homologous sequences and their structures, and then try to fit it and get identified. Also, the last is that using neural network based methods have been used to improve the accuracy. So I don't know if the CCT of text is having some error or issue, but most probably these are all the tools that can be used to identify the different kinds of sequence, different kinds of secondary, predict the secondary structure that can be helpful in further analyzing its function, its the motifs, its domains, as well as how it would be in the tertiary structure. So do you all have any question, or can try out these different tools using different structures in the different protein sequences, and can find out how they're going. So if you all have any question. Okay, so if you all don't have any question, we can conclude it here for this today's session with the protein secondary structure prediction. We will look at how the different tertiary structure prediction and all in the next session. Yeah, so stop the recording.